Welcome back. Today we are exploring Vaikrant, which is one of the most important minerals in Rasa Shastra. Now we see the mythological origin. In Puranic origin, the blood drops of Mahishasur fallen on Vindhyachal slope and which is crystallized into diamond-like shapes, so it is called as Vaikrant. Also, structural analogy, the defective fragments of Vajra are also termed Vaikrant, and pharmaceutical similarities with Vajra in Śodhana, Marana and Guna resembles. Now we see the types and varieties of Vaikrant. 8 varieties we see there. First, Śweta, Rakta, Pita, Nila, Paravat, Shamal, Kṛṣṇa and Karbura. But in Rasāyana and Rasamanjari mention 7 types excluding Karbura. We see now physical characteristic and morphology. In crystallography, 8 angles mean Aṣṭāśra and 6 angles means षट्कोण. Also present 8 facets. Smooth in touch and heavy in weight. Pure or mixed shades coloration seen. Therapeutic preferences, black variant means Kṛṣṇa Varna with 6 to 8 angles, smooth, heavy and impurity free, which is considered the ideal for Rasa Shastra use, conferring Sattva Siddhi. Now we see the Nirukti. Vikrayanti lohani rogani doshani veti Vaikrānta. Vaikrant, that is the transforms, which is lohani means facilitates transformation in alchemy. Rogani means destroy the complex disease conditions, and doshani means pacified vitiated Vata Pitta Kapha. Now we see the pharmacological and therapeutic properties. According to Rasa Chudamani, आयुष्प्रद, बलवर्णकर, अतिरुष्य, प्रज्ञाप्रद, सकल दोष गदहारी, दीप्ताग्निकृत, पवी समान, तेजस्वी, वपु बल लोहकारी. Also Rasa Chudamani Shlok 2nd, which is Rasāyaneshu, Vajrasthane means substitute for diamond, and Sarva Rogahara. According to Ayurveda Prakash, Vaikrant is a Tridoṣaghna, Shadras Pradhan, Deha Dardhyakrut, Pandu, Udar, Jwar, Shwas, Kas, Rajyakshma and Pramehagna. Also in Ayurveda Prakash, Vaikrant is explained as equivalent to diamond, also Vishagna and Rasaraj properties, and use in Kushtha and Kshaya. Ayurvedic attributes of Vaikrānta, that is Shadrasatmak, Guru and Shit Gunatmak, Shita Virya and Madhur Vipaki, and Tridoṣaghna properties. Now we see here Śhodhana and Marana of Vaikrant. According to Ayurveda Prakash, unpurified Vaikrant, which causes the toxic effect like Kilas, Daha Santati, Parshvapida and Pandu. So strictly Śhodhana is essential before its clinical use. Now we see the Śhodhana process. According to Ayurveda Prakash, first step is a Swedan. Vaikrant tied in clothes like potali and boiled in Kulathakvat using Dola Yantra. Second procedure is a Nirvap, that is heat Vaikrant as a red hot and quenched repeatedly in Ashwamutra, and same as purified with as a Vajra. Now we see the Marana process, that is a Bhasma preparation process. According to Rasa Chudamani, procedure is here: mix Shuddha Vaikrant with equal Gandhak and triturate with Nimbukswaras, prepared pellets, dry, seal in Sharav Samput and subjected to the Gajapuṭa heating. Repeated 8 puttas on an average. We make fine and bioavailable bhasma. Now we see the sattva patan process of Vaikrānta. Ingredients are here. Shuddha Vaikrant taken as a 1 pearl, that is approximately 48 gram. Tankan taken as a 1 Kṛṣṇa, approximately 12 grams. Fluxing agent 1 Kṛṣ each, that is gunja. Pinnak means sesame seed cakes and chitrakmul. Use bhavna dravya, that is a ravi kshir triturated for 1 day, and shigru swaras triturated for 1 day. Procedure detail is here. Mix all ingredients and triturated with liquids and form gutika. Dry, place in crucible. Then heat in koshti yantra for 1 ghatika, that is approximately 24 minutes, and metallic essence is released from there. Here is a one shloka for variation in sattva color. Recorded variations is white conch like sattva present, that is a jasmine or moon or conch like structure. And second variations in sattva, that is red or indragop nibham. Third is a black or iron like, that is a dark metallic iron like colors present. This is a summary. Now we see some articles related to Vaikrant. Here is first article, that is the title of Studies on Identification of Vaikrant Used in Ayurveda. Background of this study is Vaikrant is one of the important Maharas mineral described in Rasa Shastra. However, over time different Ayurvedic pharmacies started using different minerals as Vaikrant. Some use tourmaline, some use quartz, some use feldspar, and other use fluoride. Because of this confusion, there was no standard identity of Vaikrant. This could affect the quality, safety, and therapeutic efficacy of Ayurvedic medicines. Therefore, the researcher planned this study to scientifically identify the true mineral correspond to the Vaikrant. Purpose of this researcher to remove confusion regarding the identity of Vaikrant, to compare Ayurvedic description with modern scientific evidence, to establish a standard mineral for pharmaceutical use, to improve the quality and standardization of Ayurvedic medicines. Objective of this study, to collect different market samples sold as a Vaikrant, to compare their physical characteristic with Ayurvedic description, to identify their mineralogical nature using modern techniques, and to determine which mineral best matches the Vaikrant described in Rasa Shastra. Now we see the methodology. First step is a collection of sample. First step was to collect the samples. 8 different mineral samples were sold as a Vaikrant from reputed Ayurvedic pharmacy across India. Why was this done? Because different pharmacies were using different mineral as Vaikrant. Some were selling tourmaline, quartz, feldspar, fluoride. Researcher wanted to compare all of them scientifically. Now we see step second, that is a physical examination. After collecting the sample, they examined each one carefully using standard geological methods. They studied first color. Mentioned that Vaikrant may occur in 7 or 8 colors. Researcher compared the color of each sample with this classical description. Second is a crystal forms. They observe the natural crystal shape. Ayurvedic text describe Vaikrant as having 6 or 8 facets. Crystal habit helps identify minerals because every mineral forms a characteristic crystal structure. Now they studied luster, means how light reflects from the mineral's surface. Researcher observed whether the mineral had a glossy, pearly, dull appearance, which is in Ayurveda describe Vaikrant as a smooth and shiny luster. Now they study the cleavage, means tendency of a mineral to break along definite crystal plane. Different minerals show different cleavage pattern. For example, fluoride shows perfect cleavage, whereas tourmaline generally does not. Now study the fractures. If a mineral does not break along cleavage plane, it breaks irregularly. This irregular breaking pattern is called fracture. Researcher noted whether the fracture was conchoidal, uneven, or irregular. Now researcher studies the hardness using geological methods. It indicates the resistance of the mineral to scratching. According to Mohs hardness scale, quartz on an average 7 hardness, tourmaline in between 7 to 7.5, fluorite 4, feldspar 6. According to Ayurveda, Vaikrant is described as a hard mineral. Now researchers study specific gravity. Specific gravity measures how heavy mineral is compared with water. Ayurveda describes Vaikrant as a guru. Therefore, researcher measures the specific gravity using Walker's Steelyard method. Outcome of physical examination: 8 samples with Ayurvedic description, only 6 samples match the expected physical characteristic. These 6 were selected for detailed analysis. Now, third step is a microscopic examination. Now, the researcher wanted to show the internal structure because they prepared thin section of the mineral and examined them under a petrological microscope. Samples were observed under ordinary lights, polarized light, and cross Nichols. Where did they observe? They examined crystal boundaries, color, relief, cleavage, pleochroism, twinning, optical properties, isotropic and anisotropic nature. Why is microscopic examination important? Because many minerals appear similar to the naked eyes. However, their internal crystalline structure and optical properties are completely different. Therefore, microscopic examination provides more reliable identification than visual observation alone. First step is a chemical analysis. Next, researcher determines the chemical composition of each mineral. Preparation of samples. Minerals chips were washed with distilled water, crushed in a diamond steel mortar, ground into very fine powder. Fine powder ensure accurate chemical analysis. So now prepared the solution. 2 different chemical solution was prepared. First solution prepared using sodium hydroxide and hydrochloric acid. This solution was used to estimate SiO2 and Al2O2. And second solution prepared using hydrofluoric acid, nitric acid, perchloric acid. This solution was used to estimate these all major oxides commonly used to identify minerals. Now step 5 is a XRD. Finally, researcher confirmed the mineral identity using X-ray diffraction. The principle of the X-ray were passed through powder mineral samples. When X-ray struck the crystal, they were reflected by atomic planes. Using Bragg's law, the distance between crystal planes was calculated. Every mineral has a unique diffraction pattern. Therefore, XRD acts like a fingerprint of a crystal. This was the final step used to confirm mineral identity. In the first result of physical examination, among the 8 collected samples, only 6 match the classical Ayurvedic description of a Vaikrant. They probably identifies where sample first is a rubellite, second is a indicolite, fifth is a quartz, sixth is a black tourmaline scorlite, and seventh is a microcline perthite, and eighth as a fluorite. Second result is a microscopic examination. In microscopic confirm the physical observation. Researcher found that sample first, second and sixth showed optical properties characteristic as a tourmaline, and fifth showed feature of quartz, seventh showed microcline feldspar, eighth shows fluorite. Thus microscopic examination validates the preliminary physical identification. Now result of third, that is a chemical analysis. Chemical analysis shows that each mineral had a distinct elemental composition, and that in quartz containing approximately 98% silicon dioxide. This confirms that quartz is almost pure silica. Tourmaline sample means rubellite, indicolite and scarlite containing silicon, aluminium, iron, sodium, potassium, magnesium. The proportion differ among the three varieties. Scarlite compared with rubellite and indicolite had higher Si2, higher Na2O and higher K2O and lower specific gravity and hardness due to its composition. These chemical differences explain where different varieties of tourmaline have slightly different physical properties, and the result fourth of a XRD has a unique diffraction pattern. Data confirms that 3 samples belonging to the tourmaline group, 1 sample was quartz, 1 sample was feldspar and 1 sample was fluorite. XRD provided definite configuration of mineral identifier. Final interpretation of the result is after combining physical examination, microscopic analysis, chemical composition and X-ray diffraction, the researcher conclude that mineral described as a Vaikrānta in Ayurvedic corresponds to the tourmaline group with scarlite, that is a black tourmaline, showing the closest agreement with the classical Ayurvedic description. Here now we see the clinical importance. Although this was not a clinical trial, but the findings are clinically important because correct identification ensures the right raw material is used in Ayurvedic formulation. Standardized identification improves the quality, consistency and safety of medicine. It helps avoid substitute with minerals that have different chemical compositions and potentially different therapeutic effect. The study provides a scientific base for selecting Vaikrant in Rasa Shastra practice. Now we see the second article. The title is a physico-chemical study of Vaikrant Bhasma. Vaikrant is the important mineral in Rasa Shastra and is traditionally considered a substitute for diamond. Although it has been described in Ayurvedic classic, there was very little scientific evidence about the physical and chemical nature of Vaikrant Bhasma. Most quality tests described in Ayurveda, such as Rekapurnatva and Varitaratva, are subjective and depend on the observer. Therefore, the researcher wanted to validate these traditional tests using modern scientific analytical techniques. So, the researcher aimed to standardize Vaikrant Bhasma, evaluate it using both Ayurvedic and modern scientific method, determine its physical and chemical composition, and develop quality standards for future pharmaceutical preparation. Here, objectives: to prepare Vaikrant Bhasma according to classical Rasa Shastra method, to evaluate the Bhasma using traditional Ayurvedic quality test, to analyze it using modern analytical instruments, to determine the elemental composition and particle characteristic, and to establish scientific quality parameter for Vaikrant Bhasma. Now we see the methodology. First step is the preparation of Vaikrant Bhasma. The researcher first prepared the Bhasma according to Rasarasna Samucchay. First is a Śhodhana. They use black tourmaline, that is scarlet, as a Vaikrant. It was purified by heating it until red hot and immediately quenching it in Kulatha Kwat. This process is repeated by 7 times. Why? According to Ayurveda, Śhodhana removes impurities, reduces toxicity and makes the mineral suitable for therapeutic use. Second step is a Marana procedure. After purification, equal quantity of purified Vaikrant and purified sulfur were mixed. Lemon juice was added to prepare a homogeneous paste. Then leviate it. Small pellets were prepared and dry. The pellets were sealed inside into a earthen pot. These were heated using Gajapuṭa method with cow dung cake. This process was repeated 8 times until complete conversion into Bhasma. Initial weight of Vaikrant was 185 grams and the final bhasma weighted 270 grams due to incorporation of sulfur and other materials during processing. Now we see step third, that is Ayurvedic quality test. After preparing the bhasma, researcher evaluate it according to classical Ayurvedic parameters, that is a color which is observed dark brown, taste tasteless, touch results as a soft and smooth, odor results as a odorless. Nischandratva, bhasma was observed under daylight using magnifying glass. To the result, no metallic shine was seen there. And Rekhapurnatva, that is the powder was rubbed between thumbs and index finger. To result, enters the fine grooves of the skin and could not be easily removed. So interpretation is particles are extremely fine. And in Varitaratva, pinch of bhasma was sprinkled over still water. The particle floated. So interpretation, it indicates lightness and proper preparation of the bhasma. In second form, modern analytical techniques, the researcher then analyzed the prepared bhasma using advanced scientific instrument. That is, the first is a field emission scanning electron microscopy to observe the particle shape, size, surface morphology and arrangement of the particle. Second analytical technique used here EDAX to purpose determine the elemental composition of the bhasma. Third analytical technique used here FTIR to identify the functional group and chemical bonds present in sample. Fourth is a PSD to purpose determine the size distribution of bhasma particle. Smaller particle generally indicates better processing and potentially improved viability. Fifth analytical technique used here ICP to quantitatively estimate the concentration of major and trace elements such as iron, silica, magnesium, aluminium, calcium, boron, etc. In the result, the Ayurvedic test to prepare bhasma showed all classical characteristics of proper prepared bhasma, that is dark brown color, tasteless, soft texture, odorless, lusterless, past Rekhapurnatva, Varitaratva. This confirms successful preparation according to Ayurvedic standards. In field emission, same result to show granular particles, irregular shapes, uneven arrangement, agglomerated clusters, mostly amorphous or semi-crystalline particles, and particle size mainly in between 5 to 20 micrometer, which indicates the repeated trituration and calcination produces fine particles. In EDAX result to detect multiple elements like carbon, oxygen, sodium, magnesium, aluminium, silicon, sulfur, potassium, iron, boron, chromium, manganese and zinc. This demonstrates that Vaikrant bhasma is a multi-mineral formulation rather than a single elemental preparation. In FTIR result to detect several absorption peaks corresponding to different functional groups, which indicates the presence of chemical bond formed during bhasma preparation. This confirms that chemical transformation occurring during repeated calcination and processing. Fifth is a PSD, that is particle size distribution to analyze showed that 10% particle were smaller than 5 micrometer, 50% were smaller than 26 micrometer, 99% were smaller than 114 micrometer, and mean particle size was 33.8 micrometer. The researchers suggest that repeated levitation and calcination produce fine particles which may improve viability according to Ayurvedic principle. And sixth ICP result, there is a major constituent where silica is the highest concentration. After that iron, second higher concentration, and other elements detected in smaller amount includes magnesium, aluminium, calcium, boron, sulfur. Mercury was not detected there. These findings indicates that Vaikrant bhasma contains major minerals along with several trace elements. In conclusion, Vaikrant Bhasma prepared according to classical Ayurvedic methods satisfy both traditional and modern quality parameters. It is a multi-mineral formulation with iron and silica as the major constituent. Modern analytical techniques such as FESEM, EDAX, FTIR, PSD and ICP provides objective evidence that complements classical Ayurvedic tests. Combining Ayurvedic quality assessment with modern analytical tools can help establish standardized quality specification for Vaikrant Bhasma. Now we see this article for clinical importance. Although this was not a clinical efficacy study but has important implication is here to provide scientific standardization of Vaikrant Bhasma, helping ensure consistent quality. It confirms the presence of essential minerals and trace elements that may contribute to the traditional Rasāyana and Vṛṣya properties which is described in Ayurveda. Fine particle size supports the Ayurvedic concept that repeated processing enhances the potential bioavailability of Bhasma, and the study creates a scientific foundation for future pharmacological and toxicological and clinical research. Now we see third article for the title Single Dose Toxicity Study of an Ayurvedic Medicine, that is Vaikrant Bhasma. Here Vaikrant Bhasma is a herbo-mineral Ayurvedic medicine prepared from black tourmaline. It is described in Rasa Shastra as a Rasāyana and Vṛṣya. Although it has been used traditionally for centuries, modern medicine requires scientific evidence that such preparations are safe before they can be widely accepted. Why was this study done? Any medicine, whether modern or Ayurvedic, should be proven safe before it is used clinically. Classical Ayurvedic texts describe the therapeutic benefits of Vaikrant Bhasma, but they do not provide modern toxicological evidence. Therefore, the researcher wanted to scientifically evaluate whether a single oral dose of Vaikrant Bhasma produces any toxic effect. Is Vaikrant Bhasma safe after a single oral dose? Does it affect body weight or food intake? Does it damage important organs such as liver, kidney, heart, lungs, or brain? Does it alter blood parameter? What does can be considered free from acute toxicity? Here's objectives were to evaluate the acute oral toxicity of Vaikrant Bhasma, to observe animal for the sign of toxicity over 14 days, to assess hematological and biochemical parameter, to examine major organ for structural damage, to identify a dose that does not produce toxic effect. In methodology, first step is a preparation of the test drug. The researcher used standardized Vaikrant Bhasma prepared according to classical Ayurvedic methods at RGGPG Ayurvedic College, Paprola. This ensures that the test material represented a properly prepared Ayurvedic formulation. Step second, to select the experimental animals. The researcher selected healthy CF trained albino rats. Initially 20 rats were obtained, but after health screening 15 healthy rats were selected. The rats were acclimatized to laboratory conditions for 7 days before the experiment. Why acclimatize? Because the minimized stress so that any observed effect are due to the test drug rather than environmental change. And in step 3, grouping of animal. The rat were divided into 3 groups. Group first is a control group, received only 1% gum acacia to provide baseline values for comparison. Group 2, low dose group, received 10.5 mg per kg Vaikrant Bhasma orally. Group third, which is high dose group, received 21 mg per kg Vaikrant Bhasma orally. Using 2 dose level allowed researcher to detect any dose dependent toxicity. Step 4 is a observation period. After administration, all animal were observed for 14 days. The researcher monitored general appearance, behavior, activity, fur coat, eyes, mucous membrane, excreta, and mortality. Why? Because acute toxicity often appears within the first few days after exposure. So the 14 days observation period is standard for single dose toxicity study. Now in step 5, body weight, food and water intake. Body weight was measured before and after treatment. Food and water consumption were also recorded because toxic substance often cause weight loss, reduces appetite, reduces water intake. Normal values suggest the drug is well tolerated. In step 6, hematological analysis, blood samples were collected to measure hemoglobin, RBC count, WBC count, platelet count, and differential leukocyte count. Because blood is very sensitive to toxic substance, changes in this parameter may indicate bone marrow suppression, infection, inflammation, or toxicity. In step 7, biochemical analysis, to serum was analyzed for glucose, cholesterol, triglyceride, total protein, albumin, liver enzymes, bilirubin, blood urea, nitrogen, creatinine, calcium, and phosphate. Because this test assess the liver and kidney function, if the bhasma were toxic or abnormalities would likely appear in these biochemical markers. Step 8, necropsy and histopathology. At the end of the study, all animals were sacrificed. Researcher examined brain, heart, liver, kidney, lungs, spleen, adrenal gland, gonads, and gastrointestinal organ. Both gross examination and microscopic histopathology were performed. Because even if blood tests are normal, microscopic tissue damage may still occur. Histopathology helps detect subtle toxic effects. In the result, general observation throughout the 14-day study, all animals remained active. Fur coat and eye were normal. No abnormal behavior was observed. No diarrhea and no abnormal excreta occur. No death occurs, even 0% mortality in any group. This indicates that Vaikrant bhasma did not produce obvious acute toxicity. Body weight and food intake, both treated and control groups showed similar body weight gain. Food and water consumption remained comparable. This suggests that the drug did not adversely affect appetite, metabolism, or growth. In hematological result, the parameters such as hemoglobin, RBC, WBC count, platelet, differential leukocyte count remain within normal physiological limits. No dose-related abnormalities were observed. Biochemical results for liver function test, ALT, AST, ALP, and kidney function test like blood urea, nitrogen, creatinine, and metabolic parameters all remained within normal range. This indicates no evidence of liver or kidney toxicity. Organ weights and histopathology. Although few organs showed minor differences in weight, microscopic examination showed no treatment-related pathological changes. The researcher considered the finding incident rather than toxic effect. In the overall conclusion, single oral dose of Vaikrant bhasma up to 21 mg per kg was well tolerated in rats. No mortality or serious toxic effect were observed. There were no significant changes in clinical observation, blood parameters, organ weight, and histopathology attributable to the drug. Therefore, under the condition of this study, Vaikrant bhasma did not show acute toxicity in the rat. Now we see the final clinical importance of this study. Although this was an animal study, it has important implication for Ayurvedic practices. It provides preclinical safety evidence supporting the use of properly prepared Vaikrant bhasma. The absence of acute toxicity is an important first step before long-term safety and human clinical studies. The findings support the importance of classical preparation method, as properly prepared bhasma did not produce serious toxic effect in this model. The study contributes to the scientific validation and standardization of Ayurvedic herbo-mineral medicine. Now we see fourth article. The title is a comparative pharmaceutical study of Vaikrant Bhasma prepared from two different samples. Vaikrant is one of the most important Maharas and Uparatna minerals described in Rasa Shastra. However, even today there is controversy regarding its true identity. Some scholars identify Vaikrant as a tourmaline, while others consider as a fluoride, feldspar, or even manganese. This confusion affects the standardization and pharmaceutical preparation of Vaikrant Bhasma. The researcher designed this comparative pharmaceutical study because whenever there is a confusion regarding the identity of the drug, researcher compare possible candidates scientifically. In this study, the researcher wanted to compare two minerals, that is a fluorite and black tourmaline, to determine how each behaved during the preparation of Vaikrant Bhasma. Study was performed because identity of Vaikrant is still controversial. Different Ayurvedic scholars identify Vaikrant differently. Some consider tourmaline, others consider fluoride, or other manganese. Therefore, no universally accepted pharmaceutical standards exist. So, Vaikrant Bhasma has excellent therapeutic value described in Ayurveda as a Rasāyana, Vajra Bhasma substitute, and Tridoṣaghna. Yes, it is a rarely used clinically because raw material is expensive, preparation is lengthy, authentic Vaikrant is difficult to identify. So researcher wanted to know if both minerals are processed according to classical method. Do they behave similarly, or do they require the same number of putas, or do they produce similar bhasma, or can pharmaceutical observation help distinguish them? This is the central purpose of the study. Now we see the objective. We are to prepare Vaikrant Bhasma from two different mineral samples, that is a purple fluoride and black tourmaline, to perform Śhodhana according to classical Rasa Shastra, to prepare Bhasma by classical Marana, to compare pharmaceutical changes, number of puta required, organoleptic characteristic, and final Bhasma obtained, to develop a SOP for Vaikrant Bhasma preparation. In methodology, now explain each step scientifically. First step is a procurement of raw materials. The researcher collected two authenticated minerals from Gujarat. Sample first is a purple fluoride and sample second is a black tourmaline. Authentication was necessary because the study compared two different mineral identities. So step second is a Śhodhana. Because natural minerals contain physical impurities, surface contaminants, unwanted materials. According to Ayurveda, unpurified Vaikrant may produce daha, pandu, kilas, and parshopida. Therefore, purification is mandatory before therapeutic use. In procedure, both minerals were coarsely powdered, tied into a cloth bundle like Gajapuṭa and suspended in Dolayantra. Medium use was Kulatthakvat. Potali remained completely immersed. Śweta was carried out for 3 hours at approximately 100 degrees Celsius. After completion, samples were washed, dried, and stored. Kulatha decoction acts as a purification medium. Heating softens surface impurities, and continuous boiling facilitates removal of unwanted substances. This improves pharmaceutical suitability before Marana. In step third, Marana, this is the major part of this study because purified Vaikrant was mixed with equal quantity of Shuddha Gandhak and then Nimbu Swaras was added. Because citric acid helps triturate the mixture, produce uniform pellets, and promote better chemical interaction during calcination. In Chakrika preparation, the mixture was triturated for 7 to 8 hours until smooth, small pellets were prepared, and then these were dried. The dried chakrikas were sealed inside the earthen saucer. This prevents contamination and provides uniform heating. Heating was given using Gajapuṭa. Initially, lower temperature were used. Later, higher temperature were gradually introduced. Maximum temperature reached approximately 700 degrees Celsius using about 300 cow dung cake. Important observation is here for classical text mention 8 Gajapuṭa. However, researcher observed fluoride needed 11 puttas before Bhasma Siddhi Lakshana happened, and for tourmaline needed only 9 puttas to achieve proper Bhasma. This is one of the major finding. What observation were recorded after every putta, that is a weight before putta, weight after putta, quantity of gandha, quantity of lemon juice, number of uplads, temperature, color change, odor, texture, powder fineness, presence of shining materials, particles, brittleness. These pharmaceutical observation help compare both minerals. In the result, in Śhodhana, both minerals underwent purification successfully. Temperature remained around 100 degrees Celsius during Śvedana. Frothing was observed, no major weight difference according to purification. This suggests that both minerals tolerate classical purification similarly. In result second, during Marana, researcher observed progressive changes. Initially hard lump, black color, strong gandhak smell and shining particles, and in middle puttas gray color, easier grinding, better fineness, lemon smell became dominant, and in final puttas the powder became smooth, fine, lusterless and light brown to almond color, which indicates proper bhasma formulation. In number 3, number of puttas, this is the most important observation. Fluoride requires 11 puttas and tourmaline requires only 9 puttas, which indicates the pharmaceutical behavior of the two different minerals are different. In result for colors, despite starting with different minerals, both final bhasma became light brown and almond color. This shows the repeated Marana produces similar external appearance. And in result 5, that is a texture, both bhasma became fine, smooth and lusterless, which satisfy Bhasma Siddhi Lakshana. And in result 6, chemical composition, where the researcher observed only mild differences between the two bhasma. Thus, although the raw material minerals differ, pharmaceutical processing made the final products comparatively similar in appearance, nature and composition. In conclude, both purple fluoride and black tourmaline can be processed into Vaikrānta bhasma using the classical Rasa Shastra method. However, they do not behave identically during pharmaceutical processing. Fluoride requires 11 puttas, whereas tourmaline required 9 puttas, indicate differences in their response to Marana. After completion of Marana, both bhasma showed similar organoleptic characteristics, including a light brown color, fine texture, Bhasma Siddhi Lakshana, and only mild differences in chemical composition. The study also established the SOP for preparing by 2 mineral sources. And in clinical importance of here, although this was a pharmaceutical study and not a clinical trial, it has important practical value because of the standardization. Study provides standardized method for preparing Vaikrānta bhasma, helping Ayurvedic pharmacies produce more consistent medicines. Second is resolving the identity controversy by comparing fluoride and tourmaline under identical processing condition. The study adds scientific evidence to the ongoing debate regarding the identity of Vaikrānta. Quality assurance. The detailed pharmaceutical observations such as changes in color, texture, fineness, number of puttas can serve as a quality control parameter during bhasma preparation. Better clinical reliability. A standardized preparation is more likely to produce reducible therapeutic effect when used in clinical practice. Foundation for future research. The study creates a basis for future physicochemical characterization, pharmacological evaluation, toxicity studies and comparative clinical trials to determine whether bhasma prepared from fluoride and tourmaline differs in safety or therapeutic efficacy. Now we see the last article, that is exploring the concept of Vṛṣya property of a Vaikrant Bhasma. Unlike an experimental study, this is a review article. Author collected evidence from classical Ayurvedic text and available modern scientific literature to explain why Vaikrant Bhasma may possess Vṛṣya activity. Author observed that over the last several decades, there has been a decline in male reproductive health. According to modern studies discussed in the review, sperm concentration has decreased, sperm motility has declined, morphologically normal sperm count has reduced, male infertility has become increasingly common. At the same time, Ayurveda has described Vaikrant Bhasma for centuries as Vṛṣya, Vajikar, Rasāyana, Balya, and Kṣṇashak. However, there was very little modern scientific explanation for these classical claims. The author therefore attempted to bridge the gap between Ayurvedic knowledge and modern biomedical understanding. Purpose of the researcher: the researcher wanted to answer one, can classical Vṛṣya properties of Vaikrant Bhasma be explained scientifically? They attempt to correlate classical Ayurvedic pharmacology, modern elemental composition of Vaikrant Bhasma, scientific evidence regarding fertility and semen quality. Instead of conducting laboratory experiment, they critically analyzed previous literature and attempted to explain the possible mechanism. In objectives, the first primary object is to explore the concept of the Vṛṣya properties of Vaikrant Bhasma, and the secondary objectives to review Ayurvedic references describing Vaikrant, explain Vṛṣya and Vajikar concept, describe Ayurvedic pharmacological properties of Vaikrant, correlate these properties with modern scientific knowledge, explain how the mineral element present in Vaikrant Bhasma may influence male fertility, highlight the need for future experimental and clinical research. In methodology, since this is a review article, there were no patient, no laboratory experiments, and no clinical trial. The methodology consisted of a literary review and conceptual analysis. In step first, collection of classical literature such as all Brihatrayis and Laghutrayis and Rasa Shastra books, and in step second, compilation of therapeutic properties from the above granthas or classical text, which is describe Vaikrant as a Vṛṣya, Rasāyana, Balya, Medhya, Kṣṇashak, Agnidipan, Panduhar, Kuṣṭaghna, Śrotohar, and Vṛṣaghna. The consistency across multiple texts strengthen the traditional claim that Vaikrant has reproductive and rejuvenating effect. In step third, Ayurvedic pharmacological analysis, the author summarized the Ayurvedic pharmaceutical profile of Vaikrant, that is Madhur Rasatmak, Guru Gunatmak, Shit Viryatmak, Madhur Vipakatmak, and Tridoṣaghna. According to Ayurveda, Madhur Ras nourishes all 7 dhatus, Madhur Vipak enhance Shukra dhatu, Shit Virya stabilize reproductive tissue, and Guru promotes nourishment and strengthen. Based on this principle, Vaikrant is considered as a Shukravardhak and Vṛṣya drug. And in step 4, modern scientific correlation, author reviewed elemental analysis shows that Vaikrant Bhasma contains minerals such as iron, silicon, sulfur, calcium, magnesium, ammonium, and boron. They discuss published evidence on how much the element may influence semen quality and male reproductive function. As a result, because this is a review article, so the classical evidence strongly supports Vṛṣya action, because Vaikrant as a Vṛṣya, Vajikar, Rasāyana, and Balya. This suggests a long-standing traditional consensus regarding its use to improve reproductive health. And in result, Ayurvedic pharmacology supports Shukra nourishment. The pharmacological profile indicates that Madhur Ras nourishes tissue, Madhur Vipak supports Shukra dhatu, Shit Virya protects reproductive tissue, and Guru Gun promotes strength and tissue building. These properties together provides a classical rationale to improve semen quality and fertility. For modern element may explain the classical effect. The author proposed possible roles for each element, that is an iron improves oxygen transport, supports spermatogenesis and may improve semen quality. In calcium, essential for sperm motility, helps sperm capacitation and acrosome reaction and important for fertilization. In magnesium, protect sperm from oxidative stress and stabilize cell membranes, support energy metabolism. Boron influences testosterone metabolism, may improve sperm production and reproductive hormone balance. And sulfur, which is important for protein structure and antioxidant defense, may protect sperm cells. And in silicon, supports connective tissue integrity and may have indirect reproductive benefits. The author argued that these elements may work synergistically to support male reproductive health. Oxidative stress as a mechanism. Review highlights the oxidative stress damage sperm membranes and DNA, reducing fertility. So some elements in Vaikrant Bhasma, particularly manganese and sulfur containing compounds, may help counter oxidative damage and providing a possible scientific explanation for the classical Vṛṣya effect. Also, in discussion, the researcher integrates Ayurveda and modern science as follows. The classical text consistently recommend Vaikrant as a Shukra enhancement, and in modern elemental analysis identifies mineral known to support reproductive physiology. Together, these findings suggest a possible scientific basis for the traditional claims. So, the conclude here, Vaikrant Bhasma is traditionally recognized as a potent Vṛṣya and Rasāyana Chikitsa, and also Ayurvedic pharmacological principle strongly supports its role in promoting Shukra Dhatu. Its elemental composition provides a possible modern explanation for this effect. Despite encouraging theoretically evidence, well-designed laboratory studies and clinical trials are still needed to establish efficacy using modern scientific standards. Now we see the final clinical importance here. Although this review does not provide clinical trial data, but it has several practical implications to support the classical knowledge. Compile evidence from multiple Ayurvedic texts, enforcing Vaikrant's traditional role as a Vṛṣya drug. Bridge Ayurveda and modern science by linking Ayurvedic concepts with known roles of minerals such as iron, calcium, magnesium, boron and sulfur. It offers hypotheses that can be tested scientifically and guided future research to identify the need of standardized pharmaceutical preparation, physicochemical characteristics, animal studies and human trials. Potential role in male infertility. In future research confirms these hypotheses, Vaikrant Bhasma may become a valuable adjunct in managing conditions associated with poor semen quality, low sperm motility or reduced count.